Can I start? So, good morning. This is Jayashree. So, my presentation is on the topic of understanding antimicrobial resistance in the light of reactive species. So, let's start with how antibiotics act. So, it was found that usually these bactericidal antibiotics, they kill. These are agents that kill the bacteria, unlike bacteriostatics, which inhibit the growth. So, these bacteriostatics are known to create ROS formations as part of the killing mechanism. So this could be due to reduced NADPHs or the products of TCA cycle or Fenton reaction processes. So these bactericides result in surge in ROS formation in the bacteria's intracellular concentration, and that leads to cellular damages which kill the bacteria in return. So this is known as the radical-based theory of bactericide action. So, and it was found that this is common to a wide range of bactericides. So, if there is a common downstream process of killing across these antibiotics, so there should be something common in the resistance caused resistance of all these antibiotics also. So, this was the notion behind this study. So, in general, antibiotic resistance is basically a previously susceptible strain to a particular antibiotic is now not susceptible to the same antibiotic. So, that explains antibiotic resistance. So, understanding the mechanism behind this is very important as we cannot keep generating new antibiotics as it's not a feasible option. Understanding the mechanism would help us inhibit the process of antibiotic resistance formation. So, the role of mutation in development of de novo resistance is documented, but how it is done has not been previously studied. So, when we acquire these antibiotic resistance is at the sublethal dosages. That is basically when we have non-standard dosing regimens. This exposes us to sublethal levels of antibiotics for prolonged periods of time. So, what exactly happens at these sublethal levels of antibiotics is that so we'll look at how we get sublethal levels. It could be due to incomplete treatment or non-compliance with an antibiotic treatment. This all gives us exposes the bacteria to sublethal levels. So at these levels, it has been observed that the minimal inhibitory concentration increases. So even if we treat the cell line with a single antibiotic such as ampicillin, it is showing resistance to multiple other antibiotics such as ampicillin, norafloxacin, kanamycin, and tetracycline. So thus, treatment with one class of drug could lead to a heterogeneous increase in minimum inhibitory concentration. So, in some studies, it has been shown to provide a short-term protection, whereas in other studies, even after the removal of these antibiotics, long-term protection has been observed. So, since this is we can conclude that even though we treat the strain with one antibiotic and it is showing resistance to multiple antibiotics, there is again some common factor happening. So, we find out that ROS is the reason why this is happening. So, ROS production on sublethal exposure. So, when these cell lines are exposed to MIC levels of antibiotics, multiple folds increase in the ROS level has been observed. So, this increase has been proved that it's not just this correlation can be shown that the increase in ROS can lead to mutations that could give rise to resistance. So, the way ROS induces mutagenesis is that some radicals such as the hydroxyl radicals can directly interact with the nucleotides and cause mutations, or they can form mutagenic adducts with the products of lipid peroxidations. Or so when DNA damage happens, DNA repair mechanisms are induced, such as the source response. So these repair mechanisms are error prone and they lead to mutagenesis. So thus we can conclude that the ROS induced mutagenesis lead to de novo development of resistance to a wide range of antibiotics, and this is the multi-drug resistance. So in the human intestines where the drug usually is present, so bacteria, they experience anaerobic environments. So in such a case, ROS formation would not be there. In such situation, reactive metabolic byproducts of the antibiotics are formed, and these erythrophilic species, they confer killing the similar way a ROS would do in an aerobic environment. So what happens if we reduce this intracellular ROS? So certain radical scavenging agents such as thiourea and or bipyridine have been added to the cell lines to study what happens if we reduce the ROS concentration, and it has been observed that lower rates of resistance and reduced mutation rates are observed. So this again reinforces the fact that increased ROS levels lead to the mutations. And in case we increase the intracellular ROS level, so this has been experimented using single gene knockouts. These mutants, such as the OxyR mutant, which is responsible for the regulation of antioxidants, and other mutants such as the knockouts of superoxide dismutases and catalase, they all show a similar increase in the rates of resistance development. And they also show an increase in the minimal inhibitory concentrations. So from this we say that there is a tipping point in the level of ROS which will determine whether the ROS is going to kill the organism or confer resistance to the organism. And this is explained by the ROS-induced hormesis. So basically at high dosages of antibiotics for short periods of time, high amounts of ROS is generated, which basically kills the bacteria. But at the non-lethal doses for prolonged periods of time, the ROS level is lower and this causes mutagenic effects. Some of the mutations which were observed in such studies were mostly involved in the mechanisms of cell death, some in cellular processing and signaling, some in information storage and processing, and others in metabolism. So mostly the mode of action of the bactericide corresponded to the kind of mutation that was observed. And other cases, mutations of genes associated in oxidative stress response was also observed. So is antibiotic exposure the only way to gain antibiotic resistance? There are papers whereas photocatalytic oxidation. So this is basically, this photocatalytic oxidation gives oxidative stress to kill the organism. This is used in treatment of wastewater and so. But in such cases also it's been developed, the oxidative stress caused due to photocatalytic oxidation gives rise to increased tolerance of these microorganisms, which will eventually lead to formation of antibiotic resistance. So in another study where a chronic oxidative stress was induced by H2O2, was also found that there was an IS transposition, an insertional sequence transposition in a gene responsible for streptomycin resistance, for streptomyces action. So that also resulted in the resistance formation. So can we manipulate ROS to inhibit these resistant formation? So an anti-ROS agent, which was an alpha lipoic acid derivative, was added, and it was shown that in Pseudomonas aeruginosa, the resistance was inhibited, or the formation of resistance was inhibited. And in a different study, a promoter agent to ROS, such as the Fe3+, was added, which will basically increase the ROS concentration even at sublethal levels of antibiotic, which would kill the organism instead of leading it to formation of antibiotic resistance. So further studies could be done in to replicate the in vivo conditions of the human body to exactly understand how these antibiotic resistance happens. This could lead us to develop better treatment strategies so that while treating with antibiotics, we do not get the issues of antibiotic resistance. So in conclusion, sublethal level exposures of these antibiotics will lead to ROS formation, which mutates the genes in the bacteria, which would result in antibiotic resistance. So thank you. Yeah.
Who are yet to open the record? This information is too early. Go ahead. There are marks.
Actually, the last but one slide.
Yeah.
So in this, you are saying that anti-ROS agent will reverse the resistance.
It will inhibit the formation of resistance.
Okay, but like how resistance is created due to the mutation?
Yeah, yeah.
Antioxidant reagent will act only on ROS. How it will reverse the
So basically, it won't reverse the mutation. It will prevent the formation of mutation. That is what the paper says. Even before that, yeah.
quenches ROS, right?
Yeah, it quenches ROS.
Quenches ROS, but the antibacterial, like the resistance is
No. So basically, when the ROS levels are high, that leads to mutation. So they basically treat these with anti-ROS agent, which will reduce the ROS and prevent the formation of mutation, which will lead to antibiotic resistance.
The same study, like what are the controls and all this? Because the ethics that you're telling, like after treating with anti-ROS and all, it can be because of some other activities also, like the ROS in future also it can give rise to the effects in like a later phase of the cycle.
So the
How did they say like it is just because of ROS, nothing else is important in process? Like they're treating with ROS, right? In future also like you're telling
there is no future because the organism is dead by then.
the next generation.
I mean, the organism dies because there is no. If the organism doesn't die, it results in the formation of resistance.
How is it mentioned? It is just because of ROS. Like what are the controls? Like what are the
I'll have to check the paper. I'll let you know.
what happens to the antioxidant defense system when you take an antibiotic? You took, right?
Yeah, that could be.
Reactive toxin species.
Hmm.
But what happens to the antioxidant defense system? Like does it regulate the production of antioxidants?
Okay.
Also?
This has been mentioned in one paper that the innate antioxidants are not sufficient to control the ROS. That means the amount of ROS being generated due to an antibiotic, that level is not being managed by whatever antioxidant mechanism is present already in the bacteria. Yeah.
But it is the production of reactive toxin species ultimately lead to the production of antioxidant enzymes.
Yes.
So, but how it is not connected then?
Not that it's not connected, it's just not sufficient. Yeah.
You mentioned this word highest transposition.
Yeah. So,
Sir,
IS transposition.
I don't know what that is.
So basically, transposons you would have studied, right? So movable genetic elements. So that they observed that such a kind of mutation is being happened. The transposition of an insertion sequence from a different part to the gene which is responsible for streptomycin antibiotic action. So because that gene is being mutated, it results in resistance to streptomycin. Anything else? Hi. Yeah. No. So the innate immunity of the that basically it's the host's system. We are not talking about the host at all. This antibiotic resistance happens in the microbe and not in the host. So that immunity mediated is due to yeah. Yeah, but that will be, in that case, whatever is produced by the host, it is not intracellular ROS of the organism. That's basically extracellular. Yeah, it is in the environment. Yeah, in that case, the ROS produced by the host is acts as an antimicrobial way to kill the organism, but that would not confer resistance to the organism. Intracellular, yeah. Yeah. Okay. Even if it's intracellular, above the lethal level, it will kill the bacteria. It's only at the sublethal levels where the lethal level, it just means the level of ROS won't kill the organism, but it is enough to result in mutations. The thing is, it is going to damage DNA. If it is very high level, the DNA damage would kill the organism. But at that sublethal level, the DNA damage is caused, but there is also repair mechanisms. This repair mechanism is error prone, that will result in mutation. That is the link. Okay. Any other question?
Any other questions?
Thank you.
Okay, thank you, Jayashree.